morning, children. Yesterday, on Saturday, when I stopped the lecture, when the lecture was over, I was drawing the structure of endoplasmic reticulum, how the various components of endoplasmic reticulum are there. Found near the nucleus, and how they are attached. We can also call it elements. Elements endoplasmic reticulum are cisternae, vesicles, and the third one is tubules. These are the components of endoplasmic reticulum. And how do they appear? They appear like this. If we take a cell and in the cell we have the nuclear membrane, suppose we draw it here. Just like this. And the endoplasmic reticulum, first the cisternae is connected to the endoplasmic reticulum. There is a evagination, non-invagination. Invagination is this, this the when a this uh, some structure is formed, then we call it, this is evagination and this is invagination. So here evagination takes place of the nuclear membrane. Nuclear membrane protrudes out, and from there these are the cisternae.
Excuse me, ma'am. Ma'am, Annie is asking you to let her in. She's in the waiting room.
Yes, this is not visible. Yes, because my camera focus was wrong. That's why. Is it okay now? Is it okay? Is it okay? I'm speaking louder, Vita.
Yes, ma'am,
Let
is
me find
clear.
the connectivity. Yeah. So these are the cisternae, and the cisternae they have the ribosomes on it. Here, like this, we have the ribosomes on it. On the cisternae, we have ribosomes on this. These are further attached to another structure which are tubules. So tubules, as I told you, is the lumen is broader and these are branched also. So we can make it like this. These are tubules. Then we have the vesicle. Vesicles are present mostly near the cisternae, like this. Okay, and this is how it appears. So these are vesicles, these are cisternae, and these are tubules. Tubules are attached to the plasma membrane, and the cisternae, these are attached to the nuclear membrane. This is how all these structures they appear in the this is how all the structures they appear in a cell. That means to the nuclear membrane is attached the cisternae, to the plasma membrane are attached the tubules. So this is how it appears. Now we come to the next one. Is it clear, the diagram? This is how you will make the endoplasmic reticulum. Is it clear? Now I'll start with the functions of endoplasmic reticulum. Is it clear, this diagram? Okay, so I'll start with the next topic, which is functions of endoplasmic reticulum. So here functions. Number one, the functions of endoplasmic reticulum. Number one, it forms the endoskeleton or framework. Forms the endoskeleton or framework of the cell. That means, like we have an endoskeleton which gives a proper shape to a body, same way this forms the endoskeleton or framework of the cell. The second part is it transports lipids and proteins to the Golgi bodies. Transports lipids. Where are the lipids formed? Where are the lipids formed? Anybody who can answer this question? It transports lipids. Where are the lipids formed? Yes, we are talking about endoplasmic reticulum. So where are the lipids formed? Yes. Who's going to answer my question? Where are the lipids formed? I'm not getting any response. Yes, beta. Where are the lipids formed? Yes, answer my question. Where are the lipids formed? Unmute, I think. Where are the lipids formed? Who's going to answer my question? Where are the proteins formed? Yashika Bhardwaj, Amrita, Sanvi, Harsha, Siksha, Kushman, Zainab, Deepika, Jasleen, Priya, Rohan, Lalit, Sepak, Yashika. Yes. Where are the lipids formed? This is my question. Yes, I got one answer. And what is that answer? Can you send the answer again? Yes, where are the lipids formed? Endoplasmic reticulum, Deepika is saying, Sanvi Rana is also saying endoplasmic reticulum. But this is well understood that we are doing endoplasmic reticulum, but does the complete endoplasmic reticulum forms lipids and proteins both? You need to specify, you need to be very specific in your answer. You need to tell me that this is the particular part of endoplasmic reticulum which forms lipids, and this is the particular part of endoplasmic reticulum which forms, yes, Sanvi Rana is saying SER. Smooth endoplasmic reticulum. What does it form? Why it is known as smooth endoplasmic reticulum and what does it form? Yes. Why it is known as SER, smooth endoplasmic reticulum, and what does it form? Rohan, answer my question. My question is
Ma'am,
to you,
it's known
Rohan.
as Ma'am, it's known as SCR because it doesn't contain any ribosomes on its surface and it forms
Yes.
lipids.
And it forms lipids. Very good. Yes, very good. So lipids are formed by SCR, smooth endoplasmic reticulum, and it transports lipids and proteins. And where are these proteins present? These are present in RER, rough endoplasmic reticulum. They are formed because rough endoplasmic reticulum has ribosomes on it, and these ribosomes they form proteins. So both are formed in endoplasmic reticulum, and when you answer, you have to be very specific. You have to mention SER or RER. Okay, so they transport lipids and proteins. This is the second point. The third point is this endoplasmic reticulum. Okay, endoplasmic reticulum membrane, it contains about 30 enzymes. 30 enzymes are present on the membrane of ER. That means 30 enzymes are present on the membrane of ER. The ER membrane has 30 enzymes. The next point is, I told you when a cell divides like this, when a cell divides after telophase, the division is complete. The cell plate in the plant cell starts forming here, and this particular structure is known as cell plate. Cell plate is made up of few fragments. So what are these? What is this cell plate made up of? I have already told you. Who will answer my question? This cell plate is made up of, yes beta, cell plate is made up of, yes Amrita, you all are unmute. Just answer my question. Cell plate, when the cell divides in plant cell, a cell plate is formed in between. Phragmoplast. Remember, phragmoplast. Fine, it is phragmoplast. But what are these phragmoplasts? What are these? They are something. What are these phragmoplasts made up of? What are these phragmoplasts made up of? Yes. Yes. Yashika. What are these phragmoplasts made up of? Small fragments of V and high G complex. What is this? ER. These fragments are of ER and Golgi body. Sanvi Rana, very good. Yes, these are made up of phragmoplasts, which are basically small fragments of what we say Golgi bodies and ER. So the phlegmoplasts, these are small fragments of ER plus GC, Golgi complex. That means the cell, this endoplasmic reticulum is also helping in the formation of cell plate. So we can give the fourth point, helps in cell plate formation during cell division in plant cell. During cell division in plant cell. So this is what they help in, that is formation of cell plate, which is very important for the division of plant cell. Now they form an important component or important part of endomembrane system. ER forms a important part of endomembrane system. What is endomembrane system? I have already told you when we were doing this chapter. I think if you remember, you can tell me what is an endomembrane system. Anybody who remembers what is endomembrane system?
When cell organelles like endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles, they work in coordination.
This is the interrelation, like in a line, if you want to say, we can say this is the interrelationship between various organelles like endoplasmic reticulum, Golgi complex, lysosomes, and vacuole. They are interrelated in one or the other way. That forms the endomembrane system. So endoplasmic reticulum is an important part of that system, endomembrane system. This becomes the fifth point. We come to the sixth point. The sixth point is, I'll write a word if anybody knows it. Nissels granules. You know what are these Nissels granule? In which particular structure you have read this term, Nissels granule? Nissls granules. Where have you studied this? Have you
Nerve
heard this
cells,
word? Yes. Yes, beta. Speak up. Speak again. I
nerve
couldn't
cells,
hear you.
nerve
Neurons.
cells,
Yes, nerve cells. Very good. These are present in nerve cells. And you know which part of nerve cell? The cyton, the cell body part of nerve cell. The cell body part of nerve cell, which is like this. If we draw neuron, we draw it like this. If you remember, we draw the neuron like this. And here we have dendrites. Dendrites here. We have dendrites. And then what we make is this, the tail part of it, which further terminal ends are there. This is the, if you remember, this is the cyton part. This part is known as cyton part. The cyton part has a nucleus located here and it has cytoplasm. Now, along with cytoplasm, there are some, darkly colored bodies. These are Nissels granules. These are Nissels granules. These are Nissels granules. Now, what are these Nissels granules made up of? These are made with the Nissels granules which are present in the cyton are masses of Nissels granule present in the cyton. This is cyton. This is known as, this structure from here to here is known as cyton or cell body. We can call it anything, cyton or cell body. Nissels granules present in cyton of a neuron is made up of masses of RER, rough endoplasmic reticulum. This is made up of RER, rough endoplasmic reticulum. Then we have myeloid bodies which are present in retinal cells. This is the seventh point. Myeloid, myeloid, myeloid bodies in retinal cells. Retinal cells are masses of SER. These are masses of SER. So these are 7 points which are important. Functions of endoplasmic reticulum. Anything in this which you want me to repeat, or is it clear? Yes, beta. Anything in this which you want me to repeat, or it is clear? Yes, bache. Anything which you want me to repeat, or is it clear? Amrita, Rohan, yes, clear. Sanvi, clear. Okay. So this is clear to you all. I start with the next point. Next topic, which is Golgi body or Golgi complex. Okay. So I start with the next structure, which is Golgi body. Now, the Golgi bodies, these are okay. First of all, you let me know that where are these Golgi bodies present? Are these present in plant cells or animal cells? Yes. Yes. Where are these present? These are present in both, beta. So the structure, the structure of Golgi body, structure studied by Camillo Golgi. And where did he study this structure? These structures, the structure of Golgi body, he studied in the nerve cells of cats and barn owl, a special species of owls, barn owl. He was the one who studied the structure of Golgi bodies. It was in the nerve cells of Camillo of cats and barn owl. Now, there was another researcher. The name of the researcher was Dalton. Dalton. He was the one who gave the ultrastructure or electron microscopic structure. Anything you can say, ultrastructure or electron microscopic structure of Golgi complex or Golgi bodies. He was the one who gave the structure. Now, the Golgi bodies are present in both the plant cell and animal cells. Okay, present in both plant cells and animal cells. When it is present in plant cells, they are known as dictyosomes. In plant cells, these Golgi bodies are known as dictyosome, whereas in animal cells, of course, they are known as Golgi bodies. Now, these Golgi bodies, when we talk of their size, the size of Golgi body, size of Golgi bodies or Golgi complex, anything we can say, size of Golgi bodies is bigger if the cells are secretory. In secretory cells, what are secretory cells, beta? Anybody who can tell me what are secretory cells? What are secretory cells? Yes. Like liver cells. Like liver cells. Liver cells are also known as hepatocytes. Hepatocytes. Cytes means cells. Hepato means liver. Like hepatocytes. Like they are also big in another secretory cells like pancreatic cells. Pancreatic cells. It secretes pancreatic juice. So their size is always bigger when the cell is secretory in nature. And size is small in non-secretory cells. When the cells are not secretory, their size is small. Okay, like for example, muscle cells. In muscle cells, their size is small. So their size is bigger if the cell is secretory in nature, and their size is small if the cell is non-secretory in nature. Now, their number. After size, we come to their number. Their number is less in animal cells, whereas the number is more in plant cells. Less in animal cell. Their number is less in animal cell. Okay, it can be say 1 to 2. 1 to 2 only. Okay, mostly it is 1. It can be sometimes 2 and more in plant cells. More in plant cells. And in plants their number can be say 6 to 10. They can be 6 to 10 in number in plant cells. Now position. Position of Golgi bodies is they lie closer to the of course the nucleus. So this when any organelle is closer to the nucleus we call it perinuclear. When it is plants they are scattered. In animal cells they are closer to the nucleus. So the position of Golgi complex, position of Golgi complex of course we are studying. So in animal cell, if it is animal cell, in animal cell the position is perinuclear. Perinuclear, that is near the nucleus. And in plant cells, in plant cells and of course invertebrates also, they are scattered or diffused. Anything you can say, they are scattered or they are diffused. This is in case of plant cells. So this is their position in case of plants and in animal cell. Now shape, their shape. They are polymorphic in their shapes. We give the term, it is a not polymorphic, it's a pleomorphic organelle. Pleio, pleomorphic organelle. It's a pleomorphic organelle. That means it changes its shapes according to the function. Okay, so changes, that means it changes its shape according to function. Whatever function it performs, it changes its shape according to that function. So this is pleomorphic. Then one more is there, one more term is there which is associated with Golgi bodies. This is zone of exclusion. There is one more term which is associated with this, is zone of exclusion. Zone of exclusion is the area around the Golgi bodies which is in the form of a clear cytoplasm. That means there are no organelles around them. So this clear cytoplasm, which is the area, clear cytoplasmic area around the Golgi bodies, is known as this. So clear cytoplasmic area around Golgi bodies is known as zone of exclusion. Anything in this which you could not understand and or you want to ask me before I start with the structure of this? Yes? Before I start with the structure of Golgi bodies, what I told you is this structure. The Golgi bodies were first of all studied by Camillo Golgi in the neurons of cats and barn owl. Then I told you there was last point again, zone of exclusion. Okay, zone of exclusion. Zone of exclusion is where we have these Golgi bodies. Suppose these are the Golgi bodies present at one side. The area around it, it is very clear, means no other organelle is found around it. And this area which is around Golgi complex where no other organelle is found is like a clear cytoplasm, and that clear cytoplasmic area is known as zone of exclusion. Is it clear? Is it clear? Okay, then there is Dalton who studied the ultrastructure or electron microscopic structure of the Golgi bodies. These Golgi bodies are present in both, whether plant cell or animal cell. If it is a plant cell, they are known as dictyosomes. When we say the size of the Golgi bodies are bigger if the cells are secretory in nature. That means if they secrete something, if they release some enzyme, That means liver cells. Liver cells are known as hepatocytes. Hepato means liver, cytes means cells. So these hepatocytes, they release bile. So their Golgi complex is larger in size and present in pancreatic cell. Pancreatic cells, they release pancreatic juice. In the pancreatic cell also, the size of Golgi complex is big. The size is small when the cell is non-secretory in nature. That means when they do not release any enzyme. So when they do not release any enzyme, such cells are known as non